Rotam is an innovation-driven life science company operating in over 60 countries around the world. We see biotechnology as a critical part of our crop protection business, which provides solutions to farmers in all major economic crops across the planet. Working in agriculture essentially means working with live organisms. In Rotam, based on careful study of plant tissue, plant disease, insect behavior, and weed dynamics, we have developed sophisticated formulations that precisely target the problem pest. With this targeting, we can minimize the amount of chemical released into the environment. In order to control some of the world's most problematic weeds, Rotam has formed a partnership to develop a package of herbicide-tolerant seeds. This tolerance allows us to use the best available precision targeting chemicals on the crop. Best of all, this is achieved through the use of non-GMO biotechnology.
Rotam has been collaborating with trade development companies to design environmentally friendly agronomic solutions. Case in point is our partnership with Cibus Global.
Rotam has joined with Cibus and Brett Young Seeds to deliver to the marketplace our herbicide-tolerant canola, which offers a very low-cost, competitive, once-over weed control package to Canadian farmers and U.S. farmers who grow canola.
It is important to be able to create crops that are stronger, healthier, better producers, and more resistant to pests. It is our intention to demonstrate the differences between the old way to modify plant DNA, genetic transformation to create a GMO, and Cibus's new Rapid Trait Development System, or RTDS. We will address the issues with the transgenic approach and show how Cibus's RTDS system makes positive changes in plants while avoiding the issues common to transgenics. Transgenic plants are created by introducing DNA from a different organism into a desired plant. The DNA can come from other plants, bacteria, fungi, or even animals. The foreign DNA is introduced into the cell in a random location in the cell's DNA. Most likely, it will drop right into the middle of an existing gene, upon which the existing gene will no longer function normally. If it inserts into an important gene, it will negatively affect the performance of the plant. It may drop into a place in the DNA that is not expressed, so we won't even know it is there. Sometimes, several copies of the gene are inserted, which can cause expression problems. Additionally, after several generations of propagation, the gene can actually stop functioning, which is a phenomenon known as silencing. Cibus's RTDS system is a new technology that allows us to make a desired change precisely where we want, with no unexpected effects. RTDS takes advantage of a natural gene repair system which occurs in all cells and is typically used to correct DNA mistakes and mutations. It is much more precise compared to the randomness of transgenic technology and does not leave foreign DNA in the cell. The Rapid Trait Development System will use the natural gene repair system to modify a target gene to express a desired trait. In nature, there are several ways that mutations show up in DNA. One way is for mistakes to be made in DNA replication during normal cell division. Additionally, external influences like ultraviolet light from the sun can create mutations in the DNA. Too many changes would create real problems for the plant, so it has developed a mechanism to correct these mistakes called mismatch repair. It uses natural enzymes in the cell to recognize these mistakes or mutations and then repair them so the cell continues to have normal function thereafter. Cibus's RTDS system takes advantage of this mechanism to make precise, targeted changes in a cell's DNA. A gene repair oligonucleotide, or GRON, is first introduced into the cell, which pairs with the plant's DNA. Because of the specific arrangement of the nucleotides on the GRON, it attaches only at exactly the desired location in the plant's DNA. The GRON is almost perfectly complementary to the DNA of the plant, with the exception of a single mismatch, with only one of the DNA nucleotides needed to make a desired change in the gene. The plant's native DNA repair enzymes recognize the mismatch and correct the problem using replacement nucleotides found naturally in the cell and using the GRON as its template. After the repair is complete, the cell digests the GRON, which is gone within 90 minutes of introduction. The new trait is stable and segregates as any natural gene typically found in the plant. When it is all done, we have a changed plant gene that mimics natural change and variation that organisms have demonstrated through the centuries, only in a much shorter time frame. It is made up of 100% native material. None of the GRON, which was used as a template to cause the change, remains. We know exactly where the change was made and exactly what the change was. Let's look again at the issues with transgenic technology and how RTDS resolves them. 1. Transgenic technology introduces foreign DNA to the plant from another organism. RTDS uses the plant's own genetic material and repair enzymes to make precise, targeted changes. 2. Transgenic technology leaves foreign DNA in the plant. RTDS leaves no foreign DNA. The end result is 100% native DNA with a small change. 3. Transgenic technology inserts the foreign gene into an unpredictable, random, and potentially disruptive location in the plant's DNA. RTDS makes precisely targeted changes which do not disrupt expression of any other genes and which allows the targeted gene to express at the right time. 4. Transgenic technology may raise concerns with unforeseen consequences that might arise from the random placement of foreign genes into unpredictable locations within the cell's DNA. RTDS uses natural cell mechanisms to achieve precise, clearly understood, and controlled variation.
Rotam's partnership with Cibus and its non-GMO seed partner serves as an excellent example of a biotechnology solution. Our unique combined expertise of product development, regulatory, and manufacturing completes the value chain with our products, generating revenues from branded products in the significant market of weed control in canola to start with. Built upon this short-term growth opportunity, Rotam's mid-term expansion in biotechnology is primarily based on networking and investing with technology providers such as Cibus to develop new choices of agronomic solutions for more crops and geographies. The main market focus will be the Americas and Asia. Our model is evolving as we plan to participate in trait discovery and development itself to create revenue streams from both seeds and chemical solutions.
荣登对生物科技创新有着明确的愿景，制药科技的传承帮助我们成功地将先进技术应用于开发、注册和配置植物保护产品。目前，我们正步入一个新的纪元，我们正扩大与跨领域科学伙伴的合作，以便为主要农业国家的种植者以化学、生物和基因技术提供对主要农作物创新的产量和环保的解决方案。在这过程当中，我们会利用新的机遇及不断的探索与开发，来达成我们作为生物科技公司的使命，即为全人类更好的生活及环境而做出贡献，来改善这个大家共享的世界。我们会通过努力开发和应用新的技术，为我们的客户提供更好的服务，为股东创造更高的价值。